we're going to start exploring the scientific method. So instead of just giving you an experiment to do, I let you guys kind of decide what you wanted to work on today. And so our lab for today is the scientific method, designing your own experiment. So we've done safety, that should be updated in your lab notebooks. And we did the lab notebook, learning how to do that yesterday. So that should already be in your notebook as well. And so today, this is lab number 3, the scientific method. So really, what I hope that you get out of this lab is understanding what the scientific method is and using the steps to actually plan, carry out, and make conclusions about an experiment, okay? And so I hope that you'll be able to come up with a hypothesis based on observations, because yesterday you guys had to think about something you wanted to test. And so when you were thinking about that, your suggestions and ideas were probably based on observation, okay? Things that you've noticed around you that maybe you think might have a lot of bacterial contamination that you've been curious about. So you did a little bit of the design your own experimental method. So yesterday you got to think about it, but before you actually carry out your experiment today, I need you guys as a group to write out the steps of your experiment. Okay, what are the steps going to be in your process? I'm not giving you those steps, right? Because everybody's experiment is going to be a little bit different. So things to consider when you're designing your steps. What everybody's going to get is you're going to get agar plates. So those are Petri dishes. Have you guys all seen Petri dishes before? Okay, so we can take one out and show you. I'm going to get some gloves on also. So Albert has poured Petri dishes. Some gloves on, please. Oh, no, there's the small gloves. So these dishes have food for bacteria to grow. So they have sugar, they have salt. There's the correct formulation. Are these LB or? So these are, this is a formulation called Luria broth or LB. There's lots of different formulations that you can put in this plate. And it also has agar. And so it makes this kind of solid media at the bottom for the bacteria to grow on. It feels kind of like Jell-O, okay? It feels kind of like Jell-O. But you have to remember that because this is the perfect formulation for bacterial growth, that if you open the plate or you touch it with anything that's contaminated, it will grow bacteria. And so these plates have to be closed at all times. You want to minimize opening the lid for long periods of time. So Albert poured these for you guys, and the reason he has them over in that little box is that box is called an incubator. And so this is at 37 degrees Celsius, so it's warm, okay? It's mimicking a nice warm growth environment for the bacteria. And so once you do your experiment, you're gonna plate on these plates and then stick them in the incubators. So what we're gonna provide you with today is we're gonna provide you agar plates. We're gonna give you up to 3 of these. It's up to you to decide how you want to potentially section your plates so that you can do your experiment, okay? So that's that. And so one note that I want to point out to you guys is there's a lid and there's a base to these plates. Where do you think you should label these plates? On the lid or on the base? The base of the plate, okay? Because the lid rotates. It's movable. So if you write like, oh, shoe, and then over here you write phone, and you section, and then it moves, you're not going to know what's what, okay? So make sure you really look at your plate and say, okay, this big piece on top, this is the lid, okay? The piece that you can take off, that's the lid. The base is the piece that does not move. So what you're going to do is Albert is going to pass out some Sharpies. Okay, I only have a big, here's a small one. And so again, we talked about labeling. You're going to want to label your plates correctly on the base. I do not want to see you write all over the plate, because then you're not going to be able to see your bacterial colonies and take nice pictures for your results. Okay, so when you're working with plates, you're going to want to label kind of on the edges, okay, of your plate. And maybe you want to section it off. So maybe you want to, it depends on how many conditions you have. Maybe you want to separate your plate into 2 sections, and maybe you want to do a shoe and the keyboard. Okay, and so you'd have to spread bacteria on one side, that came from the shoe and one side that came from the keyboard. Maybe you want to make 3 sections on your plate. Maybe you just have 2 samples, so you don't need to make any sections on your plate. So this is up to you. That's why I'm saying your experimental design and how you do this is going to be based on your own experiment. Make sure that they're clearly labeled. If it's a shoe, you know, write shoe. Make sure that you have a date somewhere on your plate. Today's what, the 19th? Am I losing track of days? Your initials. And so see how I'm kind of writing on the edges so that you have more of the open space to look at and take pictures. Okay, so these will be your plates. So this is what we're going to give you. We're going to give you up to 3 agar plates to work with. But I'm not going to tell you exactly what to do, like as far as what goes on what plate. I want you to think about it. Okay, so materials. We're going to give you 3 LB agar plates. Okay, we're going to give you sterile swabs. So they look like long Q-tips. They're in sterile wrappers. Sterile meaning they've never been exposed to the outside air or touched on anything until you actually cut it open and pull it out of the sleeve. Okay, so they look like this. These are sterile swabs. This means that they're clean. They've never been touched by anything. Okay, so we're going to give you as many of these as you need. You need to use 1 sterile swab per sample that you're going to be testing. So don't reuse the sterile swabs. Whatever you're swabbing, make sure you get a clean sterile swab for every single sample. Does that make sense? Because then you're going to carry over bacteria. So these are your sterile swabs. We'll give you as many as you need for however many samples you have. Swabs. And then we're going to give you sterile water. It usually helps to kind of wet that long sterile swab before you start collecting your sample. Just helps pick up more of that bacteria. Okay, so we'll also give you sterile water. H2O is water, DI is distilled or deionized water. So that's what you're going to be given. Everyone's experiment's going to be a little bit different. I'm not going to give you the protocol. I will demonstrate how you should swab on your plate. Okay. Other than that, I want you to think about the steps because that's what scientists do. Okay. They need to think about, okay, what samples am I going to have? What is my control going to be? How am I going to streamline this process for all of my samples? If you're testing a shoe and glasses, do you want to swab the shoe 15 times and your glasses 2 times? No. What should you do? Same number of swabs, right? If you're going to just go in a back and forth motion, you should do that on the glasses and the shoe. So you should clearly indicate what you did. Did you just hold the swab and rub back and forth 15 times? Did you roll the swab? I mean, you want to have the exact same methodology for every sample that you're collecting, okay, to minimize variation. Okay? You want to have it streamlined. So your materials is what's going to be provided. There does need to be a hypothesis for this experiment. What is your hypothesis? It's your testable question. Okay, so you'll have your materials. Before you start, you need to formulate a hypothesis. You can generate a hypothesis with your group. You guys should talk it over and decide what is our hypothesis. A hypothesis is often, you can set it up as an if-then statement. It doesn't have to be. If I do X, Y, Z, then this is the result that I'm expecting. Okay? If I swab a clean dish towel and a dirty dish towel, then I think that I'm going to get more growth on the dirty dish towel. I mean, it makes sense. You should. When I think about, cancer research, my hypothesis was always, okay, if I treat cells with drug A, then I think they will die. Did they always die? No. Did drug A always work? No. Okay, so you have to start somewhere and come up with some type of hypothesis. So there's your hypothesis, your materials. You need to generate the method or the procedure. Okay, you need to decide on these steps. In your manual, I gave you some general steps that you could follow if you want kind of some guidance. And so I have the materials listed for you. It says Bunsen burner, but unfortunately, we're not gonna get the Bunsen burners out for this. We don't have like hardwired gas lines in this room, so I'd have to move you to another room to do that. So we're gonna just be extra careful, and I'm gonna show you how to use kind of the clamshell method to try to minimize any contamination. Honestly, even when I was a scientist at City of Hope, our lab didn't have gas. So for 5 years, anytime I did any type of bacterial work, I had to just have extra sterile technique when I was working and prevent contamination. And I had a big bottle of that LB, but it was a liquid form. I think I finally contaminated it after 4 years. But without any flame, I was able to have good sterile technique for a very long time. It's possible to do. And so here it says method, working with your group, determine your experimental design. Answer the questions below to help with your planning. You've already kind of made observations yesterday. What surfaces do you think are most dirty or most clean? What surfaces are you interested in testing, or items that you're interested in testing? You've already made those observations yesterday. Now you need to form a hypothesis and write this down in your notebook. What is our hypothesis? Maybe your hypothesis is different than your groupmate's hypothesis. That's okay. There can be alternative hypotheses to every experiment, okay? So you can write down a shared hypothesis or your own hypothesis if you'd like. Experimental design. Now you have to think, how many surfaces, samples are you going to test? What are you going to include as your control? And how will you perform your experiment? I give you a little tip. I recommend dipping your swab into the sterile water and then plating. I'll show you that example right now. Make sure you decide how you're going to collect your samples. Are you rolling your swab? Are you going to do a zigzag pattern on your swab with your swab? Are you going to go back and forth? How many plates will you need? How will you section them? And make sure you label your plates. Okay, so that's what you're going to be doing. Okay, so just to kind of refresh your memory, or maybe this is kind of the first time you're hearing this, tell me the steps of the scientific method. It kind of mimics your lab notebook entry. So what are the steps? Step 1.
Make an observation.
You make an observation, right? You make an observation, okay? Something that's happening around you, okay? So you guys made an observation yesterday. You guys have seen things, day to day, all right? What's next? You can ask a question or formulate a question, and from that question, then you will generate your what? Your hypotheses, okay? So that brings us here. There's an observation, usually generate a question. You're like, I've noticed, I personally like the Mythbuster things, 5-second rule. I'm a germaphobe, so it's really interesting to me when students do those types of experiments, like different surfaces, because my husband, right, my kid will drop a piece of food on the floor, and my husband will say, pick it up and eat it, it's fine. And I'm like, no, throw it in the trash, right? That's disgusting. So we're always having this battle.
You've never done the experiment?
I've never done it myself, but because I've taught this class so many times now that I've seen students do different variations, that I've kind of learned a little bit about it. 5-second rule, just based on what I've seen in here, it really does depend on kind of the surface of the item. Some things, like if you drop like a cracker on the floor and you swab it, it doesn't really pick up very many germs. But if you drop like a banana on the floor and it has kind of that squishy, wet, sticky texture, a little bit different situation. So I personally now feel like more on the surface of the food item, and then I also consider how disgusting is the area. Right? I mean, they did their experiments here in the lab. These rooms get mopped up, cleaned up often. So yeah. Anyway, so you will formulate your hypothesis, and then you need to actually perform the experiment to test that hypothesis. Okay, this will be what you're doing. You're going to go through, you kind of went through observation, your questions yesterday. Today you need to actually solidify your hypothesis, and you need to come up with that experiment. What are the steps that you're going to use today to carry out your experiment? And write those down so that we could reproduce that experiment if needed. Tomorrow, tomorrow's Thursday, huh? Okay. Tomorrow we'll get your plates out of the incubator and we'll see what grew on them. and we'll quantify how many colonies you got, we'll look at the morphology and see how many different types of bacteria are growing on your plates, and we'll take some pictures. Once you get those results, you need to circle back to your hypothesis. Did the results support your hypothesis? Okay? Maybe they will, maybe they won't. It's okay if they don't, right? My boss would always tell me, the data is the data. You can't force your data to fit your or serve your hypothesis. The data is the data, okay? So, yeah. And then usually once you circle back here and determine whether it supported your hypothesis or not, it helps you then think about, well, what should I do next? Maybe there was a flaw in my experimental design. Maybe I needed to swab more times, right? Maybe I should have let my plates grow a little bit longer. There's a lot that you could think about depending on what happens in your experiment, okay? So I will give you just my general example of what I've kind of gone through in the past. Question. So, I mean, this was the big overarching question of my entire thesis as a graduate student, right? I mean, the biggest question was, can this drug that I was given by a biotech company that's is being used to treat different disease, can that drug also be used to kill cancer cells? Okay, that was my job. That was my question. They handed me this drug and said, hey, figure this out. We noticed that it has anti-cancer properties, but we're using it to treat multiple sclerosis, but it seems like it could be promising for cancer. Okay, that's a big order to fill, okay? But that was my big question. That's where I had to start, okay? And so then I have to formulate a hypothesis. Okay, great, so now what is my hypothesis? After I read the literature, thought to myself, you know what? There is evidence that suggests this could kill cancer cells. But I had to get a little bit more specific. I had to think about what kind of cancer cells? Are all cancer cells the same? They're not, okay? Cancer is not all just one thing. It's, you know, every type of cancer is a very different type of disease. So I had to think about that, you know, and I had to kind of decide what kind of cancer cells am I going to treat, okay? Where am I going to start? Am I going to give cancer to an actual animal model, or do I start with cells in a dish? There's a lot of questions to ask yourself and how you're going to do that. So a simple thing that you can do is do a hypothesis with an if-then statement. And I already wrote that on the board for you guys. So, I mean, very, very generally, if cancer cells are treated with drug A, then they will die. I believe that they would die. That was my hypothesis, okay? But this was very general. I had to start somewhere. And then I had to design the experiment. Nobody told me like what concentration of the drug to use, how long to put it on the cells for, none of that, okay? So I had to start reading and come up with some ideas. So a control, remember your control is something that doesn't receive treatment, or in your case, you need to just have something that's sterile, which will just be your sterile water and your swab. Okay, something that wasn't swabbed on anything, you need to confirm that the water we're giving you is sterile, because we have a deli in the back and it gets a little grimy, and if we store tubes in there, it's possible to get contaminated. So that would be your control, but in any experiment, you always need a control. If I forgot a control on my experiment and I went and showed my boss my data, what do you think she would do? Any ideas? One, maybe she would fire me. She was a nice lady. So what she did was she looked at it, she threw it in the trash, and she said, this is a waste of time. You never do an experiment without a control because it means nothing. Yeah. I mean, she wouldn't even look at it. She'd just be like, this is trash. That's exactly what she would tell me. This is trash. You shouldn't even have wasted your time, because we cannot get anything from this. If there is not a control, you shouldn't have done the experiment at all. So yeah, it's a rude awakening when you maybe forget to do that control and you're so excited and you have this data and you're like, look at what I did, and they're like, nope, you did it wrong. But I never made that mistake again. You know, as a young scientist, you might forget a control. You might be so excited about your experiment that you didn't think about the proper control, okay? And so yeah, I learned that lesson and I never did it again, ever. Because the reaction was very negative and very harsh, which was deserved. It was deserved. And so experimental group, This is gonna be identical to the control group, except they receive the treatment or they receive whatever you're testing. In your case, you're gonna use a new swab, you're gonna dip it the same way, but now you're going to actually swab something, okay? You're gonna collect a sample. So that will be your experimental group. In my case, my experimental group was the cancer cells that were gonna get the drug. My control was cancer cells that were not gonna get the drug, but were gonna get whatever liquid the drug was made up in. So if my drug was made up in water, that's what it was resuspended in, then the cells got the same volume of water that my experimental group got. Does that kind of make sense? So you never know what effect water could have on your cells. If you made it up in a different solvent, that can actually be detrimental to the cells themselves. These two words always get students very confused. Dependent and independent variables. So maybe I should just have you look at that without telling you what it means. Do you guys have any ideas? What is an independent variable? What's a dependent variable? Talk to one another and see if you know what this means. What is the difference between those two things? And even if you don't know, just look at the words and see what you think. Go ahead.
So independent, it acts on its own, does its own thing. Dependent, it gets a reaction. Yeah, no, I'm the same way. It's like, it gets, like even like my face and breathe, it gets sweat.
Better close the door. They always tell me I'm too loud. You're too loud. Close the door. I want to hear you. Okay, so tell me, what is an independent variable? And what's a dependent variable? Any ideas? What did you guys talk about? We'll start with, I don't know, either one. Does anybody have any examples or want to put it in their own words?
Isn't dependent and that's dependent on the variable that you're testing?
Yes, okay, so that's that's a good way. It's dependent on something that you're doing to it. It's gonna be something that could potentially change in response to whatever treatment you're doing. Yeah, so it does depend on whatever you are doing or interacting with. What about independent? Just the word itself. Does it depend on anything that you're doing? No, usually the independent is something that you choose ahead of time. There are variables, but you are in charge of those variables to start with. So let me give you some examples. These are kind of fixed values that are already known before the experiment, and they don't change. You decide on them before the experiment starts. So let's give some examples of independent variables. So they're fixed, meaning they don't change when the experiment starts. You decide on them ahead of time. So what would be an independent variable for my cancer cell experiment? Any ideas? What type of cell, right? Okay, type of cell. What else? What else do you need to know ahead of time? That would be fixed. What was that?
What drug you're using?
The drug, absolutely. You'd want to know what drug you were using. What else do you want to know about the drug? That would be fixed before you start.
Concentration.
Yes, you'd want to know those concentrations, okay? So the concentration of the drug. Concentration. What else? How about how often, or maybe, so this is the drug concentration that you're going to use. What about the time frame? How long are you going to treat for? Okay, treatment time. And we might even, with treatment time, you may even want to decide on your collection points as well. Because do you just want to give a drug and take one time point 2 days later? Is that going to be useful? If it works, they'll probably all be dead and you won't know anything about the kinetics of the drug. So usually what's really interesting is treatment time. Maybe you want to do 48 hours total, okay, that'll be your last time point. But you might also want to know time points within that 48 hours that you want to collect you want to collect information about the cell viability. So maybe collection times. So maybe this would be something like 12 hours, then you're going to look again at 24 hours, and then again at 48 hours. Okay? So these are all independent variables. These are all things that are fixed. Usually the researcher or scientist decides on them before the experiment starts. Okay? They're already kind of fixed and planned out. Okay? In this experiment for my cancer cells, what's going to be the dependent variable? I told you that I wanted to look at cell death in response to drug treatment. So what's the dependent variable? Go ahead and talk to each other. What's the dependent variable in my experiment?
Zero positive. It's very flexible. For the actual data.
So remember, this is fixed. And then this will change, or it could change. Okay, so dependent variable. What's the dependent variable that I'm looking at here? What are we looking at? What did I say I wanted to look at? I want to look at cell death. So what am I going to be evaluating at 12 hours, 24 hours, and 48 hours? How many cells are alive or how many cells are dead? You can graph that different ways, but you're still looking at cell viability, okay? So yeah, you could look at cell survival or cell death. Those tell you, right, two sides of the same coin. Cell survival or cell death. Because that's what will depend on all of this, right? And the cell survival might look different at 12 hours, it might look different at 24 hours, and it might look different at 48 hours, right? It's dependent on these independent variables. So it's what will change, and it usually is impacted by what you decided here, right? If I picked a concentration that was too high, maybe they're all going to be dead by 12 hours and you have nothing left to look at. If I pick a concentration that's too low, they might not die at all, and then I might think, oh, this drug doesn't work. So it's tricky. You have to really think about it. And so yeah, this is the data these are the results that are only known after the experiment happens and as you're collecting. So in the cell survival case or the example that I was giving you, you're going to look at cell survival or cell death, okay? And so this is, these are the examples, I've already written them on the board. Control group, cancer cells that didn't receive treatment. The experimental group is the group that's going to get my drug or drug A. Independent variable, I put time, but here's many other independent variables that go into the experiment. And then this could be either cell survival or cell death, depending on how you want to graph it out and look at it, okay? And so here are some sample results. Okay, this would be a typical graph that I would make after I did an experiment. So looking at this experiment, what can you say? What information can you gather? Did my drug work?
Yes.
Yes, how do you know? Because you see that at the very beginning, at time 0, okay, the cells were 100% alive because I'm plotting percent survival. How many of them are alive at that time point? They were alive. Not only my cells that I was going to treat, but also my control cells. So you always have to make sure the cells are happy and alive. Because what if one dish was super angry and contaminated with bacteria, and then the other dish was very happy? I mean, and so then right when you start out, there's something wrong. My boss would always tell me if your cells aren't 95% viable or higher, don't even do the experiment. Okay, something's wrong with the cells. Okay, and so as you can see, my control at all the time points stays alive. Is that what you expect? Absolutely, we didn't do anything to them. We just gave them food and let them grow. Okay, and you can see with drug A that at 24 hours, 48 hours, 72 hours, this was a longer time frame, that you saw a nice decrease in survival over time, okay? And so what's the conclusion? Did this piece of data support my hypotheses? Yes, it does. If we treated with drug A, these specific cancer cells did die, okay? If you look at the graph, when data is presented to you, where do you usually find the dependent variable? The x-axis or the y-axis? So look at the graph and think about this. It's the y-axis, because remember, our dependent variable was cell survival, and here it is, cell survival on the y. That's usually how it's presented. And then here, the independent variable, this was time, okay? And sometimes we'll even write concentration as well, depending on what was done. So yes. So independent is on the y, dependent is, independent is on the x, dependent is on the y. All right, and so that's there. So now it's your turn. So here's just a little example. Make sure you label your plate on the bottom, okay? As a very general protocol, remember that you're gonna wet your swab and spread. So I wanna give you a very brief demo, and then I'm gonna let you plan your experiment and do what you need to do. Okay, so let me get my gloves on, and I'll show you how to do this. And if you're unsure or you need help, Albert and I will be walking around, and we can help you guys and kind of direct you. If you're unsure about your experimental design, you can run it by one of us before you start, just to make sure you guys feel 100% confident with what you're doing. Okay, so you're gonna wanna get lab coats on, your gloves on, proper PPE, pull your hair back because you are gonna be spreading on a plate. You've already cleaned your bench tops off, so that's good. Sterile water is gonna be in this rack at the front of the room, okay? So I'm gonna write control on this side. This will be my control, which is gonna just be water alone. Okay. So again, do not open your plate at all until you're absolutely ready to start spreading. So you're gonna want to kind of prepare yourself, and you don't wanna open this water and leave it open for the elements to drop in there. So what I do is I kind of open my cap so it's loose, and I can work with it a little bit easier. So what you're gonna wanna do is you're gonna wanna open your swab, feel where the tip is. You don't wanna tear at the wrong end. You don't wanna grab the swab from the cotton end. You wanna feel and open it at the end where the little stick is. You're gonna pull the swab out, do not touch it on anything. Sometimes I see students put it on the desk, they touch it on their lab coats, they just are not paying attention. Don't touch it on the outside of the tube or the outside of the plate. That's a no-no, because then you're picking up bacteria from those locations. Okay, so what you're gonna do is you're gonna remove the cap, go straight in, dip, and you can even kind of take a little bit of the excess off, put the cap back on, don't touch it on anything. Okay, you don't wanna be talking, I'm talking because I'm giving you a demo. Then what you're gonna wanna do is what we call the clamshell method. You open up the plate like a clamshell to prevent anything from falling in there. See that? This is enough, just a little clamshell. So I'm gonna position the plate so that it's my control side. Should I come to the middle or can you guys see me? I need like a little camera on me. Okay, so I'm gonna open clamshell. You're gonna wanna place your swab kind of at a 90-degree angle, okay? And you're gonna wanna just gently rub back and forth, okay? Back and forth, back and forth. Just kind of be cognizant. I would probably swab whatever surface you're working with kind of right at the tip and on the side, and try to rub that same area on your plate so that the bacteria transfers onto your agar. You don't want to swab the back side and then you're swabbing with the wrong side of your Q-tip and then you're like, oh, no bacteria grew, okay? Because maybe none of them transferred on there. This agar is like a jello. If you push too hard, it's just gonna rip. So it's a little bit of a finesse. So again, clamshell, I do not want to see this. I don't want to see plates open and you're over here dipping in and you're taking a while and you know, you touch it on the outside, that's a no-no. So you definitely, clamshell, lay it on there. It's hard to do it holding it in my hand. Don't hold it up like this, I'm only doing that so that you can see. You're gonna wanna swab gently back and forth and then immediately close the dish, okay? This is now used, okay? So you don't wanna use this one again, you're gonna discard this. This you can just throw in the trash can. There's nothing that you're picking up that wouldn't already be there, okay? So you can just throw this in the trash can. When you're all done plating, you're gonna wanna flip your plates upside down, okay? And if you have a stack of 2 or 3, we have this colorful tape here in the front. You're gonna wanna make sure all your plates are upside down, stack them together, and tape them up, and put them in the incubator, because what always happens is people steal other people's plates, or when they're taking their plates out, they drop them and they open, so we don't want any of that to happen, okay? Any questions? So upside down, then you're gonna walk over here once they're all taped up. This is the incubator. You're gonna place them in upside down. You're gonna place them on the shelf upside down, and you're gonna close the little door. If you see the stacks in here, all the plates are upside down. Any idea why? Why shouldn't we put them right side up? Yeah, condensation, because it's a warm, humid environment in there, and you could even see, that's why I dropped the plate. There's actually some condensation on the outside that kind of ran down the sides, so it's a little bit wet right now. Just condensation. Water will kind of go up to the top, and then it'll drip all over your bacteria. And so instead of getting nice little colonies, it'll just look like a soup, and it won't be nice. Okay, so make sure you flip the plates upside down, so the lid side down. Okay? All right, so go ahead. I'll leave that up there, and you guys can get started. If you need to go out and collect your samples, go ahead and do that. Just make sure that you get lab coats on. If you're gonna wear gloves, like if you're going somewhere disgusting, like a restroom or something like that, I'm okay with you wearing gloves, but there are a few rules. If you're gonna walk out of here, out of that door with gloves on, you can only have 1 glove on at all times. Make sure you are using your ungloved hands to touch doors, okay? You should always take 1 glove off if you're walking in public. You should never have both gloves on. That's just a practice that you should always use. And you don't want to forget and use this hand on the door. You always want to touch doors with your ungloved hands, okay? You can put your other glove on when you get there, or if you want, you can take gloves in your pocket. Put your gloves on when you get there, but on the way back, make sure that you have a glove off so you can touch doors, elevator buttons, any of that, okay? So we don't want people freaked out. And it's just sanitary, germaphobe, good practice. Oh, another thing, when you're taking your gloves off, let me put my gloves back on, because you might go somewhere gross, and I just want you, and I'm touching them, so this is actually, I'm gonna give you a good demo right now. I'll put them on. I know I didn't touch anything, so I should have more care. So I'm gonna do this. Okay, if my gloves are clean, they're on. Okay, great. Maybe we should do this next time. I know another professor that gets food dye, and he like sprays food dye all over the students' hands and makes them take their gloves off without getting dye on any of their arms or fingers. It's a good idea. Okay, gloves are on. How should you take your gloves off? So one way would be to grab it and pull. Okay, pull it off so that the dirty side is now inside out, okay? And it's always good to kind of get your finger in underneath, right? So you're not touching the dirty part, and pull it off as well, okay? So you can kind of pull off and put them in the trash can. Now they're both inside out, so if I touch them, whatever was on your hands is like, you know, the inside, your hands should be clean. I would also go wash your hands before you start this off, just to be on the safe side. Make sure you wash up after you're done. Make sure you clean your bench tops off after you're done. And make sure you throw all of your trash away. Okay? All right, have at it. Go ahead.
Did you guys want to use trash can in the bathroom? Because I don't know if you throw things into it. It's very dirty. Do they have lids? Do they remember, or are they like that?
I think they're like, well, we could
Oh,
do
silent.
like right where the bag like ends, you know what I mean? On the outside maybe. Or I think they're like the ones in like the metal things. We could do those.
Oh, actually, there's a trash can on the corner, and it has like a circular thing, and you know how like when you throw away something, it touches the bladder, and then it goes into the little yeah. So we got the trash can, we said pencil.
So you wanna do like all of it?
So here's the sign-in sheet. It's going to be going around. Please make sure you sign, and I'll collect it at the back.
If you guys have any ideas,
No.
Well, what's the
Putting
hypothesis?
I'll show you what I did.
What do you wanna know?
Honestly, I fully thought
Yeah, out of that experiment.
that's fine.
I'm gonna move the gloves to the front. If anyone wants to come and practice, kind of swabbing with my practice plate, you're more than welcome to try that. If you wanna just give it a practice before you do your own plate, I'll leave it up there for
So
you.
like we can just bring it in. It's like
When you're collecting sample, I would, yeah, just put gloves on
you
so
could
that
just write
nothing
all
that
those
was on your
different
hands
objects to
gets
see
on
which
your
one's the most
hand.
like
And it's just good laboratory practice.
You know what I mean?
Wear gloves.
and then let's start
Get a lab coat on.
Right.
I don't know what made me think it was like right there.